We're starting our last chapter on everyday chemistry. In this case, we're going to be talking about two very important biomolecules, DNA and proteins. And we're going to start with DNA and how it stores and transfers genetic information. DNA stands for deoxyribonucleic acid. It's a polymer that carries all the genetic information in all the species that we know of, and it contains the information to make a full corn plant all the way to a human being. The DNA molecule is very flexible, as we'll see later on. It could replicate really easily, transfer the information faithfully, and then respond to feedback from within the cell. And that response is really important because it tells the DNA molecule to only translate certain information that is stored within it and not other information. The way information is stored in the DNA is in a series of what we call nucleotides, which is a molecule that's composed of three chemical units: a nitrogen-containing base, which is shown here, a deoxyribose sugar, which is shown here, and a phosphate group, which is shown here. The sugar and the phosphate look the same for all DNA molecules, but there are four different bases in them. The one shown here is called adenine, but there's also another one that's somewhat similar called guanine. And then there are smaller bases called cytosine and thymine. A typical DNA molecule has thousands of these nucleotides combined together through a covalent bond that's called a phosphodiester bond. The formation of that bond is shown here. We have one nucleotide here, we have another nucleotide here, and what happens is the OH group in the phosphate group of one nucleotide reacts with the hydrogen from a neighboring sugar from another nucleotide. Then they form this bond, that's the phosphodiester bond, and now you have a dinucleotide, or two nucleotides combined together. Repeating this again and again and again form this single helical structure, which is called a single strand of DNA, where this ribbon that's colored in blue is what we call the sugar phosphate backbone of the DNA. And then these little horizontal bars represent the different bases that stick out from that sugar phosphate backbone. Now it turns out that the bases can make hydrogen bonds with each other. Because of those hydrogen bonding, that results in a three-dimensional pattern that we call a double helix. The hydrogen bonding pattern is very specific, and that's because the larger base, adenine, can hydrogen bond specifically with thymine, and then the other larger base, guanine, can hydrogen bond with cytosine. So you always have an AT pairing, and then you have a CG pairing. So in this picture right here, you see the specific colors are always paired up together, like green with yellow, and then the red with the orange. They're always paired together, and that's because those are the specific pairing of the bases. So the yellow and the green are cytosine guanine pairing, the red and the orange are the thymine adenine pairing. That immediately tells you that if you have a specific sequence of bases showing in one strand, automatically the other strand would complement that first strand with the correct base. The reason we know that the structure of DNA is this double helical structure was through a combination of two techniques. One is an experimental technique called X-ray crystallography, and the other one is model building based on that data. X-ray crystallography is a technique that's illustrated here. What you need is a crystal of a molecule, in this case a crystal of DNA. And then when you shine an X-ray beam on the crystal, the molecules inside that crystal will diffract the X-ray, which just means to scatter it. This is similar to when you shine a flashlight on an object and then project that on the wall. You see that parts of that light will get shine on the wall, and then parts of the light will not, because it's blocked by the objects. Very similar idea there. With the molecules in the crystals, we can look at a pattern called a diffraction pattern on a film, and based on that pattern, deduce what the molecule might look like. The person who actually took that X-ray picture of the DNA crystal was Rosalind Franklin, which is shown here. And this is the actual diffraction pattern of the DNA that she took. Just from the look of the cross pattern in the X-ray picture, it was immediately clear to people who knew about X-ray diffraction pattern that this is a two helical patterns, very similar to when you have two coiled springs. These two arcs on the top and the bottom is what suggests the stacking of the base pairs. So if you look at the structure of the DNA, the bases which are pairing with each other are also stacking on top of one another, thereby giving you this pattern right here. So this is one pairing of a base, this is another one, but they're stacked on top of each other. Because of these two experimental evidence, two other scientists, James Watson and Francis Crick, built a physical model. This is the model shown here. This is Watson, this is Crick, showing that this particular model of the DNA, which it's a little hard to see, but it's a double helical pattern where there's a base pairing of each strand, is what gives rise to this X-ray diffraction pattern. Now, it's unfortunate that only Watson and Crick and a third person, Morris Wilkins, who's the supervisor of Franklin's work, were awarded the Nobel Prize, which is sort of the highest award you can give to a scientist. She herself wasn't honored with the Nobel Prize. And later on, there's a lot of arguments that she should be awarded that Nobel Prize because she was the person who took that picture that was used to build the model. The double helical structure of DNA explains a lot in terms of why DNA can store and transmit genetic information. So the information is stored in this double helical pattern in the mother cell, but when the mother cell divides into two daughter cells, the DNA also unwinds just like this. And so there are enzymes in the cell that allows this to happen. So enzymes are biological catalysts. They make chemical reactions happen in the cell. So the enzymes would unwind the DNA to make them two single strands. And then another enzyme would come in and help to synthesize two new strands of DNA. So if you see here, this is the blue DNA is the one from the mother cell. And then this golden colored one is the new one that is starting to be synthesized. The sequence of DNA that is synthesized would be based on what's present on the mother cell, but it's complementary. So for example, if you have a sequence of ATT on the mother cell, then on the daughter cell, that corresponding sequence that would be synthesized would be TAA because it would complement what's shown on the mother cell. Similarly, on this side, you'll have that TAA from the mother cell. Now the daughter cell would synthesize ATT. So now you have two copies of exactly the same DNA strands that originally appear in the mother cells. Now you have two copies of it, one in each daughter cell. And that's how we get that same information transferred from generation to generation with fidelity. Fidelity here refers to the fact that it has very little error. There is some errors that are made by this enzyme, but very little error that you're able to make the same information again and again and again through generations.